This video is about the lateral geniculate nucleus. The lateral geniculate nucleus is in the pathway from the retina, lateral geniculate nucleus, to the cortex. So it's like a switching station, and it's part of the thalamus. When you look at the lateral geniculate nucleus, it actually has 6 layers. And they're in a certain order. Layer 1, Layer 2, Layer 3, you'll see why I'm drawing them these different colours in a moment. Layer 4, Layer 5, and Layer 6. 1, 2, 3, 4, 5, 6. But there's an order to these, and that is that the layers 1, 4 and 6 come from the contralateral eye, whereas layers 2, 3 and 5 are from the ipsilateral eye. And that's because when you look at the optic nerve, let's imagine that we've got 2 retinas here, I'm just going to redraw that. So here's a retina. I've drawn it in 2 halves. So that's my contralateral eye, and I'm going to draw my ipsilateral eye retina in 2 halves again, because what happens is that the axons from this part of the retina pass along the optic nerve and stay on the same side and come out on the same side of the brain, but the axons from this part of the optic nerve cross over to the other side of the brain. And it's the same with this eye, with the ipsilateral eye. This side does not cross over and stays on this side of the brain. This side does cross over and ends up on the opposite side of the brain. And this happens, this part here is called the optic chiasm, because it's like a Greek letter chi, which is an X. So the result is each side of the brain now has both ipsilateral and contralateral fibres, but they represent the same part of the eye, because you'll notice if you trace these back, this covers this part of this retina. If you trace this one back too, it covers the same corresponding part of the retina. So the receptive fields overlap, but we've got information from the left and right eye. This now ends up in the appropriate layers of the lateral geniculate nucleus. So what we got here is another example of parallel processing. The information is being brought to, I'm in trouble spelling, I don't think that's right, is being brought to the lateral geniculate nucleus from both eyes, registered by where they are in the visual field, but the streams are still kept separate. But there is actually a slight complication with this. There's another dimension to all this, and that's got to do with going back to the retina, because the retina the output from the retina, the retinal ganglion cells, there are two types. There's the parasol retinal ganglion cells, and there are the midget retinal ganglion cells. So remember, these are small receptive fields, and these are large receptive fields. So these are give rise to what we call the magnocellular pathway, and these give rise to the parvocellular pathway. It's really annoying that we've got for M to P and P to M, but magno and parvo are easy to remember. And it turns out that when you look at the route that these take, that, just trying to find my window again, the parvocellular, the magnocellular pathway goes to layers 1 and 2. So these are all. So this is where the magnocellular pathway goes, and then the parvocellular pathway goes to these layers. So that means that P cells actually go to layers 1 and 2, and M cells go to 3, 4, 5, and 6. So just one final note from all this is to think about the actual output of these. And once again, we find that we have this parallel pathway taking place. So the output from the magnocellular layers goes to the cortex, but goes to they all go to layer 4C, which is the input layer of the cortex. These go to 4C alpha, which is a part of the 4C layer, and whereas the magnocellular pathway, sorry, I've got that the wrong way round. The magnocellular pathway goes to 4C alpha, the parvocellular goes to 4C beta. So alpha and beta, I'll just rub that out for you. So the parvocellular goes to 4C beta, and the magnocellular goes to 4C alpha. So that then is a very rough, approximate overview of the lateral geniculate nucleus.